Hello, my name is Guillaume Ramstein. I'm a postdoctoral researcher at Cornell University. And today I'm going to talk to you about how we can predict evolutionary constraints using genomic annotations and how we can use those in functional enrichment with applications in genomic prediction, especially in Maize. As you probably know, in genomic prediction, we used a whole set of markers along the genome, and based on effects of those markers, we predict a particular phenotype of interest. But ideally, we will want to use only the subset of the markers that have an actual effect on the phenotypes, those variants that are causal. But we cannot do that. But however, what we can do is use genomic annotations to prioritize the subset of markers that are more likely to be actual causal variants. And we can use those in genomic prediction later. So the approach that we use to prioritize markers is to first estimate the effect of some types of markers, specifically single nucleotide polymorphisms, on fitness. And we predicted SNP fitness effects by genomic sequence through genomic annotations. And the genomic annotations that we used described the SNPs with respect to genomic structure as well as protein function, with annotations at the level of genomic regions or at the level of individual loci. For example, mutation effects, like whether a SNP is a missense mutation or a stop-loss mutation, for example, as well as CIF scores. But we also used genomic annotations which describe the protein ontology, that is, a quantitative representation of the protein structure and function based on neural networks. And we use also disruption scores, which described how that protein ontology was changed as a result of the mutation. And for SNP fitness effect, what we used was actually a proxy through evolutionary constraints, which described the conservation at each individual site in an alignment across 27 plant species, not including Maize. And then to learn the relationship between the functional annotations and evolutionary constraints, we use a typical machine learning model, random forests. And so then we wanted to validate the functional relevance of our predicted evolutionary constraints. And so we compared predicted evolutionary constraints with measures of within species conservation, which was not included in our model. And we observed, as expected, a strong negative relationship between predicted evolutionary constraints and minor allele frequency, which reflected within species conservation. And an interesting result as well was that that relationship was stronger than what we could get directly from the observed evolutionary constraints. Also, there was a significant increase in predicted evolutionary constraints in regions in the genome that were open, either in root or in shoots. And we did not observe such significant differences in constraint for the observed empirical evolutionary constraints. And finally, we wanted to assess whether our predicted evolutionary constraints were useful in genomic prediction application. And so here I'm showing the prediction accuracy that you could get in a particular validation scheme for a grain yield in maize through random weighting of SNPs, and as a result, random prioritization of those SNP markers. And what you observed is that when we used predicted constraints from our model to prioritize the SNPs in genomic prediction, we could get a significant increase in prediction accuracy compared to those random prioritizations. And so this method that I described based on functional enrichment could be useful in genomic prediction applications, but they could be also useful for selecting a set of candidate variants to then improve a plant variety through techniques based on molecular design, such as genome editing for many loci simultaneously. And I would like to end this presentation by acknowledging my collaborators in the Berkeley Lab at Cornell University, as well as my funding sources.